Hello, and welcome to our third Rare Disease Day webinar. I'm Professor Matthew Wood, Director of the Oxford Harrington Rare Disease Centre and Professor of Neuroscience at the University of Oxford. We have a wide range of speakers today to tell you more about our work in rare disease drug development and in the new opportunities in the global rare disease landscape. Thanks very much for joining us today. So we have a packed agenda for you over the next hour. I will give the introduction with my colleague, Jonathan Stamler. We then have a number of messages from stakeholders, academic research stakeholders, and others. We will then follow this up with my colleague, Diana Wetmore, and take questions and answers. And we will conclude with some closing remarks from Jonathan Stamler. Now, the rare disease challenge that we face today affects 400 million people globally. Many of these people suffer from severe rare diseases, and this means that approximately 1 in 17 of all people on the planet are affected. More than half of these rare disease patients are children. Now, there's been enormous government support across Europe and the US over the last 30, 40 years to support rare disease drug development. 40 years ago, the Orphan Drug Act was signed into US law. And in 1983, there were 38 drugs approved for rare diseases. And there are now approved therapies for over 1,000 rare diseases. In the UK, a UK rare diseases framework in 2021 sets out a vision for improving the lives of people with rare diseases and promoting personalized medicines. So with these enormous advances and with the scientific advances in genome sequencing, programmable personalized genetic therapies, we're in a position now to make enormous progress for the huge unmet need in the vast majority of these diseases that do not have a treatment. And in order that we do so, we will be able to benefit the millions of people who live with rare diseases. Now, our strategy for doing this is built on partnerships. The partnership between the Harrington Discovery Institute in Cleveland and the University of Oxford is the partnership that underpins our entire effort. The Harrington Discovery Institute has funded more than 150 drugs in the making, of which 40% are already in rare diseases. And through these efforts, it has delivered 31 spin-out companies and 13 licenses to pharma, and it now drives therapeutic development across the US and also in the UK. The other half of the partnership is the University of Oxford, which has enormous critical mass in rare disease scientific research, with over 250 researchers working on translational research projects in hundreds of rare diseases. And its development and capabilities in innovation therapeutics have been enhanced by the recent COVID-19 challenge. So together, this is the partnership that is building our enormous rare disease effort. This partnership is being brought together in what we have called the Oxford Harrington Rare Disease Centre to deliver our vision, mission, and goals in rare disease therapy development. And on this slide, these are stated. Our vision is combining excellence in research, based in Oxford principally, and drug discovery, which is provided by the Harrington Discovery Institute to cure rare diseases worldwide. Our mission is to deliver medicines to patients suffering from rare diseases. And our goal, our explicit goal over the next 10 years is to deliver at least 20 new drugs for rare diseases to benefit the 400 million people worldwide who are suffering from rare diseases. How do we do this in practice? So this is our therapeutic development focus and strategy represented on this slide. We've built this around the expertise that we have in Oxford and at Harrington Discovery Institute to focus on 3 critical areas: neurological disorders, developmental disorders, and cancers. Rare neurological disorders comprise the vast majority of all rare diseases. Possibly up to 70% of all rare diseases have at least a contribution in the brain. Our efforts are built around three areas: neuromuscular rare diseases, ataxias, and rare epilepsies. The ataxias is a great example where we've made progress recently, building on our earlier efforts in neuromuscular disease. And we built the Friedreich's Ataxia Alliance at Oxford to focus immediately on developing therapeutic programs for Friedreich's ataxia. In the developmental disorders space, we have recently built a huge partnership with the British Heart Foundation, bringing in significant investment to focus on rare heart muscle disorders. And we're busy right now putting together partnerships and investment to support our research activity and drug development work in rare cancers. Our strategy in all of these areas is to accelerate the development of drugs for rare diseases to get to the point where we have at least 20 of these drugs in clinical trials in 10 years. And that acceleration strategy is built around bringing the expertise in drug development and aligning this with the financial investment we need to deliver the 20 therapeutics. I'll now hand over to Dr. Jonathan Stamler, President and co-founder of the Harrington Discovery Institute, to update us on their recent activities. Thank you, Jonathan.
Thank you, Matthew. I'm Jonathan Stamler, President of the Harrington Discovery Institute. I'm very happy to celebrate Rare Disease Day with you. Harrington was formed in 2012 to accelerate promising discoveries into medicines for unmet needs. We do this through 4 innovation centers: a center for major diseases, one for brain health medicines, one for COVID-19, and a 4th for rare diseases. Among those 4 centers, our Rare Disease Center has tremendous momentum, gained through a strong and growing partnership with University of Oxford. Oxford has 250 rare disease researchers, one of the best assemblies of rare disease scientists in the world. With a commitment to advancing breakthroughs, regardless of where they originate, our Oxford partnership has great promise to find cures for children. For our part, we have a phenomenal team of therapeutics development experts who specialize in research, drug development, regulatory matters, clinical trials, commercialization, licensing, company formation, and more. Together, we are accelerating the development of therapeutics for rare disease and improving outcomes for our patients. The momentum I mentioned comes from these combined strengths, as well as our forward motion. On that front, we are actively launching the Oxford Harrington Center Accelerator. This is a dedicated, patient-focused rare disease accelerator that will match the top discoveries with elite translational capabilities, in turn with funding opportunities ranging from grants to commercial investments. The purpose of the accelerator is to carry promising therapies from the US and UK through clinical trials and into approved medicines. We think this is an exciting source of rare disease capabilities and funds. Through the accelerator, we intend to initiate dozens of clinical trials. The accelerator is thus an important source of new capabilities and funds to help counter the economic burden of rare disease. Because the reality is that rare disease drug development is expensive. My job is to find, promote, and underwrite new models to address great need and show that not doing rare disease drug development is far more costly. There is, in fact, staggering cost to not developing treatments for rare disease. We are making inroads into all of this. To date, at Harrington, we have received 650 rare disease applications and seen 242 top-tier discoveries. We now have 43 rare disease medicines in the making, 6 companies launched, 4 rare disease pharma licenses, and 2 rare disease medicines in the clinic. With that said, I'm very pleased now to turn things over to Diana Wetmore, our Vice President of Therapeutics Development. She'll share examples that illustrate our breadth in translating discoveries into new treatments. Thank you.
Thank you, Dr. Stamler, for that great insight into innovations underway and the future of rare disease treatments, both at the Harrington Discovery Institute and at the Oxford Harrington Rare Disease Center. Harrington Discovery Institute deploys a unique model in fulfilling our mission to accelerate promising discoveries into medicines for unmet needs. Our model is to discover, advance, and cure. Novel discoveries are sourced from premier academic centers leveraging Harrington's transatlantic network of scientists and partner institutions. Harrington advances innovative science through the drug development process by providing extensive pharma know-how through its Therapeutics Development Center. Drug development experts de-risk academic programs and help advance them into the clinical realm. Intellectual property rights remain with the scholar institution. And Harrington support cures by engaging industry and investment partners who are able to commercialize new medicines and bring them to patients in need. I will now provide some examples of our Harrington model in action through 3 of our rare disease projects. I think they show the momentum we have gained in delivering treatments for rare disease. First, Justin Uchida. Justin Uchida is a Harrington scholar focused on treatments for amyotrophic lateral sclerosis, or ALS. ALS is caused by repeat expansion of part of a protein, which causes the protein to clump, leading to the death of nerve cells. Dr. Uchida has found that inhibiting the levels of the PIK5 protein blocks this clumping. His technology has been licensed to the company Accurastem, and it is in the preclinical candidate stage. Dr. Jeannie Lee is working on the treatment for Rett syndrome, a rare chromosomal disorder that occurs almost exclusively in girls. Symptoms become evident at about a year old and include loss of strength, seizures, absent speech, and autism. Individuals typically live wheelchair-bound and require full-time care. Females have 2 X chromosomes, one of which is dormant, and Rett is caused by mutations on a gene within one of the X chromosomes. Dr. Lee's aim is to alleviate disease by reactivating the normal gene on the dormant X chromosome to restore the gene's function. The technology is called X chromosome reactivation, and it is incredibly innovative. It is in the lead to candidate stage. Ed Grabczyk is a Harrington scholar working on Huntington's disease, which causes brain nerve cells to waste away. Symptoms typically appear in middle age and include uncontrolled movements. It too is caused by a repeat expansion. In Huntington's, the repeat expansion is found within the Huntingtin gene. Dr. Grabczyk found that the repeat expansion gets longer over time, accelerating the onset of the symptoms. This is caused by a kind of side activity of the MLH3 protein. Dr. Grabczyk is attempting to halt the repeat expansion using a clever approach where he can splice the MLH3 gene to get rid of that side effect activity and thereby halt progression of the disease. His approach also may enable treatment of other DNA repeat disorders, such as Friedreich's ataxia. Dr. Grabczyk's technology is in the lead optimization stage. And now, I'm pleased to introduce you to Dr. Angela Russell from the University of Oxford, where she is Professor of Medicinal Chemistry in the Departments of Chemistry and Pharmacology. Professor Russell is a Harrington UK Rare Disease Scholar, who is also the first scholar supported by the Oxford Harrington Rare Disease Center. Professor Russell will present on her work advancing new drugs for the treatment of Duchenne muscular dystrophy. She'll be followed by comments from her lead Harrington advisor, Dr. William Greenlee. Dr. Greenlee is an expert in medicinal chemistry with 35 years' experience in drug discovery and development. whose work has led to the approvals of multiple new drugs. He formerly served as Vice President of Cardiovascular and CNS Chemical Research at Schering-Plough and held leadership roles at Merck Research Laboratories. First, we will hear from Professor Russell, and then she will be joined by Dr. Greenlee following her presentation.
Thank you, Diana, for the very, very kind introduction. So I'm going to talk to you in the next 10 minutes, a little bit of the background of the work that we've been doing on Duchenne muscular dystrophy, some historical work, and how that led to the collaboration with the Harrington Discovery Institute and what a difference that's made to what we've been able to do. So Duchenne muscular dystrophy is a relatively rare condition, although it affects around 1 in 3,000 to 5,000 boys. And it's a really debilitating condition. So it's typically diagnosed around 2 to 5 years old when muscle weakness begins, but it's a progressive disease and affects all muscles used for movement, as well ultimately as those for breathing and in the heart. So typically children with DMD, therefore, are confined to a wheelchair, usually about 12 or in their early teens, and it dramatically reduces the average life expectancy to the 30s or early 40s. And at the moment, there's no cure for this disease. So there are some therapies, although they that have been recently approved, but they show very limited efficacy and are only restricted to certain patient populations with certain types of this disease. And the reason this disease is such a challenge is it's caused by one of hundreds of different gene mutations that we know, which prevent these children with the disease from being able to make a protein called dystrophin. And so if you look at this figure at the bottom, on the left-hand side, this is a biopsy taken from the muscle of a healthy individual. And what you're seeing stained in white is this protein dystrophin at the membrane. And on the right-hand side, you can see a comparative section from a patient with Duchenne muscular dystrophy. And what's obvious is it's characterized by this complete absence of this protein. And what this protein does is it acts as a shock absorber in the muscle, preventing these muscle fibers from being damaged during contraction, so in normal exercise. So if it's absent, you get then this damage and degeneration of the muscle, which is progressive. So our approach to this disease is to try and develop a therapy that increases the levels of a protein, which for several years ago, Professor Dane K. Davies, with whom we've had a long-standing collaboration, showed could compensate for this defective protein in the disease. It's very, very closely related to dystrophin, and you can see this image here with the two proteins lined up side by side. And several studies have shown that utrophin has the potential to act as a substitute for dystrophin in all children with DMD. So now, importantly, this could function irrespective of the underlying genetic mutation to either stop or slow the progression of the disease. So just this figure here shows zooming in on an individual muscle fiber and looking at that complex at the membrane where dystrophin is located. And so what we're aiming to do here is to reconstruct that link, which is missing in DMD patients, by replacing it with an alternative but similar protein, utrophin, and thereby restore the integrity of the muscle and the function. And so the question we asked is, could we identify a small molecule, orally delivered drug, to increase the utrophin levels to treat Duchenne muscular dystrophy? And so we set out to do this almost 20 years ago. And over the years, we were able, from our work and then together with Summit Therapeutics, to take the first drug designed to treat Duchenne muscular dystrophy by increasing the amount of utrophin in muscle. And this drug was called ezetimibe. And we took it into a clinical trial with Summit Therapeutics back in 2017 in boys with DMD. This trial was designed to ask the question as to whether this would improve muscle structure, muscle health, and increase utrophin. And what we saw after 24 weeks of treatment was really encouraging. And this is the data that you can see on the right-hand side of this slide. So at the top, you can see a section from one of the patients at baseline, and all of those individual muscle fibers, which are stained in blue, are expressing a protein called developmental myosin. And this is a marker of degeneration and regeneration. So that is indicative of a significant amount of damage in the muscle of this particular patient. And then at week 24 underneath, you can see a biopsy from the same patient after 24 weeks of azutrigumab treatment. And visibly, there was a reduction in the amount of damage in that muscle. And when we looked at this across the cohorts, there were about 40 children on this study, we could see the statistically significant and meaningful reduction in the level of this particular marker of degeneration in there. And that correlated with improvements in inflammation and fat fraction in the muscle and other markers of inflammation and damage. And we saw utrophin increasing alongside this. So this looked incredibly exciting after 24 weeks. However, when we looked at the end of the trial at week 48, it was as though everything had reverted to baseline. We could see no difference between the children that had been treated with this drug compared to the natural history, the natural progression of the disease. And so it wasn't effective over a longer period of time. And that led in 2018 to Summit discontinuing the development of azutrigumab. And it left us in Oxford with a question to try and understand why was there no sustained benefit of azutrigumab treatment. And so over the next 2 years, we set about to address this question. And the first thing we sought to understand was the mechanism through which this drug was acting, because we didn't actually know the details of that at that time. And so we've used a range of techniques, and through these, we've been able to demonstrate that azutrigumab binds to and blocks the action of a nuclear protein called the aryl hydrocarbon receptor. So this was predominantly done through the technology that's shown on the slide here, where we converted azutrigumab into a photoaffinity-based probe, which is the molecule shown here, the little blue bar at the top, where we can incubate these with live muscle cells and then irradiate and cause that molecule to capture any protein in which it's in close proximity. And then through a process of breaking open those cells and conjugating that probe, we can then understand and fish out effectively the target through which azutrigumab is binding, and therefore work out the mechanism through which it's operating in these live muscle cells. And so that's what we managed to figure out. At the same time, we looked into understanding what happens to azutrigumab when it's administered in these children. And we knew that azutrigumab undergoes a metabolic process to these two metabolites shown on the slide here. And we also knew that these are inactive or very, very weakly active as in increasing utrophin levels. And so this is effectively an inactivating metabolic process. But what the problem was, and we noticed this a little when we looked at healthy volunteers in the early stage clinical trials, was not the metabolism, but that the more that we dosed azutrigumab, the levels of the drug were falling with repeat dosing. And this happened, as is shown here on the right-hand side, even over the first 10 days of treatment in healthy volunteers, but it happened to a much more significant extent when we looked at them in the trial with the children, which we were not expecting. So this fall in azutrigumab levels means that at the beginning of the trial, you have enough drug in the children to engage utrophin, but over time, the levels, the therapeutically effective levels of this drug fall away, and then that leads to a subsequent but slower drop in the utrophin levels, and can explain why the benefits that we saw midway through went away with a longer treatment. And so this was where we were. We had to halt the work on azutrigumab, but by understanding how this works and why it didn't give this lasting benefit in the clinical trial, it actually allowed us to uncover a new opportunity to look for now alternative drugs that could act in a similar way, but would be more effective and provide long-term increases in utrophin. And this was really where, back in 2020, we put the application into the Harrington Discovery Institute, and I was very grateful to be awarded a scholarship in order to address this question, build on the findings that we had so far, and try to really use this to accelerate the development of the new molecules towards the utrophin replacement that would have lasting benefit in patients. So this is where we were when we began our work with Harrington. And we were very fortunate Harrington, the funding from Harrington funded a chemist in my lab to be able to start to synthesize new molecules that would allow us to generate new molecules to take us forward with the project. At the same time, we received funding in the UK from Action Medical Research that allowed us to support biologists on the project in parallel so that we could establish a pipeline to make and test molecules to identify better molecules that we could develop into the clinic. But it wasn't really about the funding. That's been extremely helpful to us. It's about the partnership that we've had with Harrington that's been so instrumental in the progress that we've been able to make. So at the bottom, you can see on the left is Kay and Matthew Wood, with whom we work on this project in Oxford, but also Bill, who you're going to meet in a minute, and we'll talk about the project, Ashley, Joe, and of course, Diana, who has already spoken, introduced me, and Mike Hallen, who's responsible for coordinating the project. And it's really been this expert advice and this support that's made such a difference and allowed us to move so quickly. So in the course of a year, we were able to identify new chemical matter, these ligand-selective aryl hydrocarbon receptors, which we showed were active in vitro. And that in turn, we were able to trigger some stage 2 funding from Harrington, which allowed us to really accelerate the pace at which we were working. And this has been tremendously helpful to increase our progress towards our goals. And that allowed us over the course to generate a proof of concept molecule and optimize next generation antagonists. And that in turn, in December 22, triggered and leveraged a larger increase in funding from the MRC in the UK, which is really, again, allowing us to build critical mass, accelerate the progression of this over the next 2 years, and where we're aiming in 2023 through to the end of 2024 is where we're eyeing being able to progress this into the clinic. So this is where we're going. So I've explained already what we've been able to achieve since our partnership began with the Harrington Discovery Institute to be able to discover and optimize these alternative molecules as AHR antagonists, which increase utrophin production. So we're anticipating these compounds will allow us to overcome the limitations of azithromycin, and we're going to be developing those into a new candidate drug in the longer term for the long-term benefit of all children with Duchenne. And we're going to be able to do this because of this funding that we've just received from the UK Medical Research Council, which will allow us to take these into the next phase of drug development to ensure they're safe and effective. So in the next stage, I'm going to introduce Dr. Bill Greenlee, who's been instrumental as a medicinal chemistry advisor on our program. And I think we're going to have a chat about how we've been working together.
Thank you, Angie. I am Bill Greenlee, the lead HDI advisor on Professor Russell's DMD program. I'm a medicinal chemist and been working with HDI scholars for the past 10 years. I wanted to start our discussion by just saying a few words about our collaboration. We've been working together on the project for the past 2 years and are continuing into the first half of 2023. This has really been an outstanding collaboration, and I've really enjoyed my interactions with Professor Russell and the entire project team. It's really been a unique scholar program for me. Professor Russell is the first Oxford Harrington rare disease scholar that I've worked with, and she's a medicinal chemist like me, so it's been really a different experience from the other scholar programs that I've supported. Professor Russell mentioned DMD as a devastating disease that starts early in life. So there's a large unmet medical need for treatments, especially for small molecule drugs, which is what we're focused on. And we believe that the path that we're taking is the best one to treat all DMD patients. Our HDI team members really share the determination that the Oxford group has to identify an effective small molecule to treat this disease. As Professor Russell mentioned, the HDI team members include Ashley Rowe, Jan Rosenbaum, and our HDI project manager, Mike Hallam. And many thanks to Diana Wetmore for her support and encouragement. Last year, HDI provided stage 2 funding to the DMD program, and that allowed us to accelerate the synthesis and evaluation of new small molecules in a new lead series. So we've been able to make a lot of progress during the last year. And what's really exciting is that our most recent compounds now are over 100-fold more potent than earlier leads in the program and have favorable drug-like properties. So our goal for 2023 is to identify potent inhibitors for additional in vivo efficacy studies and to try to make significant progress towards a development candidate that will be suitable for clinical trials for DMD. for me, this has really been the most enjoyable and exciting scholar program that I've been involved in. And I'm grateful to you, Angie, and all of the DMD team members for an outstanding collaboration. And I'm really looking forward to seeing the program succeed. So Angie, I'd like to turn it over to you for some brief comments on the Scholar program.
So as I mentioned as I was going through my slides, thank you, Bill. That's very kind, and the feelings are mutual. I think it's been a fantastic opportunity to interact at all levels. So it's so much more than just an investment of cash. I mean, it came, of course, at a critical time for us in that we'd just figured out what we thought had gone wrong with the clinical trial, with a hypothesis that we wanted to be able to test for how we could actually achieve lasting benefit. But it's so much more than an injection of cash at the right time. The interaction with you, the interaction with Ashley, and then Jan, in fact, was brought in later to help us with the program when it was obvious that really we're dealing with a nuclear receptor. So somebody both with the insights into the business development, but also experience on working with nuclear receptors was going to be beneficial to the program. And that flexibility and that breadth and depth of expertise and the way that in which we can interface with that has just been tremendous. And no problem's been too big to overcome, right? It's been an amazing experience. You know, we have a problem with unusual PK. Somebody that has an expertise in PK is brought to the problem. The crystal structure of the aryl hydrocarbon receptor is suddenly published. We suddenly have an interaction with a computational chemist who can help us to try and derive information that will help us in the design and optimization of our molecules. I mean, it really that flexibility and that real breadth of expertise and the ability to call on these people and have them work so well together, I think is I've never come across it anywhere before.
Thank you, Angie. Yeah, you've mentioned some of the challenges that the program faced, and that actually was one of the questions that was posed for us for the discussion. And I thought about the chemistry also. We're doing some chemistry that's really unprecedented in the literature. And we've actually, since we have 3 medicinal chemists on the project team, we've been able to steer around some of the roadblocks there and actually get this chemistry to work. And it's actually been kind of enjoyable to work on some new chemistry and develop it. So that's been great. And the other thing I'll say is that the aryl hydrocarbon receptor is a difficult target. There's some challenges there. And I think we've really put in place a careful analysis of new compounds that we're making to make absolutely sure that they're going to work and they're going to be able to help patients in clinical trials and beyond.
I would agree with that. And I think also the planning process of thinking exactly what do we want our molecules to look like and what do we not want our molecules to look like, and that expertise that we were able to bring in to address that question has been hugely valuable to do. Yeah, I completely agree.
Okay, thank you very much, Angie, and thanks for a great collaboration.
Thank you very much.
We can now move on to hear from our global rare disease advocate, and we're very fortunate to have Giulia Vitarello with us here today. Giulia will share her remarkable journey as Mila's mother, and also as an advocate for accelerating rare disease therapeutics more generally, and bringing together the global networks needed to take on this enormous challenge. Giulia has established Mila's Miracle Foundation and is also a key participant in the N of 1 Collaborative. And these endeavors are established in order to support this rare disease challenge. Giulia and Mila's story concerns an ultra-rare form of Batten disease, a devastating genetic neurological condition. Thanks so much for joining us today, Giulia.
Thank you for inviting me to speak at this year's Rare Disease Day. Before I tell you a little bit about my daughter, Mila, I wanted to show you this short video just to get a chance to get to know her.
You are my sunshine.
So our daughter's name is Mila Virginia Makovets. She was born on November 5th, 2010. When I think about Mila as an infant, I think about smiles and laughter and health and...
Half an hour after she was born, she was strong enough to hold on to my finger and I could almost pick her up off the bed.
When Mila was a toddler, she was a climber, she was a hiker, she was a runner.
How old are you, Mila? Here's our sugar high daughter.
Slide, slide, slide. Woohoo!
Very physical, always loved to ride a strider.
Nice, Bug.
Play in the snow, go sledding.
I'm coming!
We noticed that Mila was getting stuck on her words. She would say, "Mommy, Mommy, I have to. Mommy, I have to," and not be able to continue. And then suddenly, around 3 years old, we started noticing that her feet were slightly unturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way, and then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. You know, we took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She could no longer see anything. What's brought her so much happiness is playing with her toys and or looking at her books and watching TV and playing with her brother, and suddenly it looked like she could no longer play with anything. It's so hard to see such an outgoing little girl not be able to play with other kids and not be able to play with toys and not be able to watch Elmo anymore. As you can see, my daughter and my son were completely healthy. You know, I was like any other family, but my daughter started to have all of these problems. And I was told by over 100 doctors and therapists over the course of 2 years that I was a crazy mom and that my daughter was completely normal. After 2 years of being told that, I gave up. I had dragged her around the US and into Canada, and no one really could figure out what was going on with her. And they thought she would grow out of these symptoms. I brought her in after 2 years to the ER, and I just gave up. I put my hands up, and I hoped that they would figure out what was going on with her. And after a series of tests, she received one of the worst diagnoses any child could ever receive. She was told, and we were told, that she had a rare form of an already rare disease called Batten disease. And I went from first feeling enormous relief that I wasn't a crazy mom to spending, about 2 seconds on the internet only to see that Batten disease was one of the worst diseases out there, and that Mila would lose every one of her abilities and die in the next few years. My life as I'd known it kind of ended on that day. But I picked myself up. I started reading white papers. I started learning from other families who had fought for their children. And I started a foundation because that's all I could do. I started telling my daughter's story and raising money and working towards a gene replacement therapy. But there was a problem, as my daughter was missing a mutation and she needed 2 in order to confirm her disease diagnosis. And so in that quest to look for this other missing mutation, I turned to social media, and I needed to get her into a lab in Boston, which was one of the only labs in the country at the time that did this whole genome sequencing, which was needed to hopefully find this missing mutation. I needed that in order to test my son, who was very normal, just like Mila had been. And I thought, motherhood was going to be ripped from my arms. And I looked at my little son every day, who at the time was, 3. And I thought he was going to start falling and tripping and stuttering just like Mila had. And I also needed that in order to make sure that I was going after the right gene. And that if I was raising millions of dollars towards a treatment, that it was going to be for the correct disease. So in that process, I ran across Dr. Timothy Yu, who stood up and said, let me try to help you, with his team from Boston Children's Hospital. And in just a period of 1 year, they not only found that my son did not have Batten disease, thank goodness, but also came up with an idea for a medicine that would be tailored to Mila's mutation, which they did find. This was kind of a crazy notion at the time, because Mila was the only person in the world that had this mutation, which meant that the drug that they were developing would only be for her. So over the course of that year, everyone raced against time. And 1 year later, we moved to Boston, and Mila began receiving this antisense oligonucleotide, or ASO, called Milasen, which was named after her. And, you know, in the first year, Mila did incredibly well, despite the fact that she was losing her abilities rapidly over that year. She had lost her vision. She had lost her ability to talk. She was losing her ability to walk. Her seizures were going from, you know, none to 30 a day in just a period of months. And as she began receiving Milasen, you know, despite really being at a progressed state, she was still smiling and laughing. But with Milasen, you know, her seizures pretty much disappeared. There were days she had none. Sometimes she had just a few. She started sitting up much stronger and taking better steps with my support from behind. She started eating by mouth again. You know, and most importantly, as a mom, she started laughing and smiling a lot more than she had before. And, you know, that year was amazing. It was kind of hard to believe that my fight, you know, and my fight with Dr. Timothy Yu and his team at Boston Children's was able to actually show a really meaningful change in Mila's life and our life as a family. And the second year of Milasen was not as promising. It was unclear whether or not it was stopping her disease or not. And in the third year, her disease continued to progress. And 2 years ago, my daughter's big spirit left her little body after a really hard fight. And I am here today still talking about this because Milasen was not in time for my daughter, but it showed what was possible. It showed that a drug targeting the underlying genetic cause of disease, even if it's unique just to one person, can be made and can be administered. And I didn't go into this, you know, trying to make my daughter the first person in the world to receive a drug tailored to one person, but that's what happened. And it showed the future of treating disease, especially rare genetic disease, where little to no hope exists at all. And so I go to bed at night thinking about the 60 million children who will die before the age of 5 from a rare disease. And that's the population of Italy. And that's where I lived all my 20s. And I can't go to sleep at night thinking about this because it feels like a global health crisis. I don't know why no one uses those terms. And right now, we're barely able to help any of those children. But the approach that was used for Mila could be used across rare disease as a whole. And it could offer an incredible solution, very impactful solution. But the problem is that our system was not designed for this. So our system was not designed for, you know, it was designed for one drug for tens or hundreds of thousands of people, not tens or hundreds of thousands of drugs, each for 1 or 2 or 6, you know, dying children. And so I spend my days through my own foundation, Mila's Miracle Foundation, through the N of One Collaborative, which myself, together with Timothy Yu, who is one of the Harrington scholars, and has greatly been supported by them in order to continue this work. We have continued working forward to try to make sure that this individualized medicine approach, you know, is taken into consideration in how we treat genetic disease, and that we start to change the system and change the framework that we think about developing drugs for rare disease. And I'm very grateful for the Oxford Harrington Rare Disease Center for playing a huge role in this and for helping make sure that more and more children are treated and that we learn from them and that we pivot and we improve and then we continue treating more and more children to open up this path. The Oxford Harrington Rare Disease Center offers, you know, huge hope for children like my daughter. And while it wasn't in time for her, there are millions more that we need to help. And so I'm very grateful for all the support and thinking of my daughter, especially, and the millions like her out there today on Rare Disease Day. Thank you.
Thank you, Julia, for sharing your family's story and the tremendous work of Mila's Miracle Foundation. Now let's move on to our questions and answers portion of the program. If we don't get to your question today, please do email us directly at info@oxfordharrington.org. Here's our first question that came in. The therapeutic development approach we learned about today was a small molecule. Is the Oxford Harrington Rare Disease Center also progressing advanced therapeutics towards the clinic? For example, oligonucleotide drugs and gene therapies. This is a good question for Professor Wood. Professor Wood, will you please share your thoughts?
Thanks, Diana. I mean, this is a great question. One of the key parts of our strategy is to be agnostic as to the therapeutic modality. So we are entirely comfortable working with small molecules and also with advanced therapeutics. And we cannot know in advance which modality will be the most applicable for each rare disease that we tackle. But having said that, advanced therapeutics is a critical element of what we will need to do. The rare disease disorders that we're taking on are genetic, and many of these will require a genetic therapy, a gene therapy. And this is a key part of the work that we're doing. For example, we've already been successful in developing oligonucleotide drugs, and we've recently secured significant funding from the UK government to establish a UK-based oligonucleotide research capability, and in our own team, new technologies for the delivery of oligonucleotide drugs to the brain and other tissues that we will need. So these developments will significantly underpin our therapeutic efforts, and we aim to build parallel therapeutic efforts in gene therapy as well as small molecule drug development so that we can deploy whatever the appropriate therapy is for each rare disease.
Okay, thank you. Next question is, can you talk about where the intellectual property of a scholars project lies in the Harrington model? Does it belong to Harrington Discovery Institute or the scholars host institution or someone else? I think I can jump in here. The Harrington Discovery Institute does not take any intellectual property rights to the projects we support. Those rights belong with the scholar and the scholars institution according to their own internal protocols. Next, there's so many rare diseases, over 7,000. How does the Oxford Harrington Rare Disease Center decide which rare diseases to focus on? Professor Wood, this is a good question for you.
Yes, I mean, this is a very, very important question. And I mean, this just emphasizes the scale of the challenge that we have. We don't really know how many of these diseases there are. You've said over 7,000. Some estimates suggest that it could be hundreds of thousands, and that underpins the urgency of our work. Our initial approach to this is that the disease has to be rare. It has to be genetic so that we have a clear therapeutic target. And most of the diseases that we take on will be pediatric, affecting children, and will be severe, where we believe we can really make a difference to the patients and their families. Beyond that, we've really thought to prioritize our activity and resources on those diseases where there is the greatest need and where we have the expertise to advance drug development as rapidly as possible. Hence, our focus on the brain, neurological rare diseases, and rare cancers as being two of the biggest challenges that we're going to take on over the next 10 years.
And one more for you, Professor Wood. The Oxford Harrington Center has ambitious goals. What needs to happen in the funding landscape to achieve these goals?
Yeah, this is a critical question. I mean, as we outlined earlier in the webinar, our strategy for the future really is to accelerate rare disease drug development. And that brings together the right expertise in drug development and the financial resources to accelerate the development of drugs. So the fundraising landscape and the financial landscape is actually absolutely critical to what we need to do. I think the investment to support our efforts will come from numerous sources. We know and have highlighted earlier how governments are aware of the huge need for rare disease drug development. And certainly in the UK, there's enormous government investment into the infrastructure and increasing investment into the infrastructure needed for rare disease drug development. But we need more than that. We need industry, we need the investment community to be playing their roles. And I think many in industry and in investment can see why rare diseases are so important, not just because they benefit the patients suffering from these diseases, which is absolutely critical, but because advances in rare disease drug development can lead to advances in the developments of treatments for more common diseases. The other element of future fundraising is philanthropic support. And this is absolutely critical for us. And I think there really is now a recognition on both sides of the Atlantic, but particularly the US, which has a much longer tradition in this area, that philanthropic support can absolutely underpin all of our efforts in rare disease drug development, allow us to take those early stage risks in projects to generate the data that will subsequently lead to large pharmaceutical collaborations or large scale investment. So all of these different sources of investment are going to be critical. And I think there really is a mood and a recognition that now is the time to do this, that we can do this, and that the funders and investment community are ready to support this effort.
Okay, this will be our last question. Is there any chance that a rare disease therapy, for example, the one we heard about today, that it may be used or adapted to treat other similar rare diseases? I'll first defer to Professor Russell, and then I'll ask Professor Wood to comment on the focus of work funded at Oxford and how we create impact in rare disease.
Yeah, it's a good question. So I think it will depend on the specifics. So in the case of ours, where we're trying to increase utrophin and trying to increase stabilization of that complex at the membrane, there are other diseases where that might be relevant. But I think more generally, the strategy that we've taken, the idea of increasing levels of a compensatory mechanism or activating a compensatory pathway in order to treat a disease where there's a genetic deficit is definitely tractable. So I think there are learnings and things that we've developed as part of the work that we've done that would be applicable in other conditions. So for instance, we've also been interested in trying to apply this sort of approach in Friedreich's ataxia, where there's a similar idea that you could increase levels of the frataxin protein in that context and use that as a treatment for the disease. So I think that most certainly there's an opportunity, at the very least, to take these principles and apply them into other disease contexts to treat other rare diseases.
Yes, thank you, Professor Russell. I mean, let me just build on the comments you've already made. I mean, this is a great question, and this is really an important one for our Rare Disease Centre. Because there are so many rare diseases, and we're aiming to develop at least 20 new treatments, we want to make sure that the treatments we develop have the greatest impact possible. And so where possible, we envisage that the 20 drugs we develop will not just treat one rare disease, but could treat multiple rare diseases. So yes, the answer to this question is absolutely yes. We are looking at our rare disease drug development pipeline from exactly this perspective, trying to identify therapeutic opportunities and diseases where one drug and one drug development effort could benefit multiple diseases and multiple groups of patients. And this could happen in a variety of ways, either by technology that we develop for one being broadly applicable to many other diseases, or in some cases by the very same drug that targets a particular biochemical pathway or element of the cell being directly applicable from one rare disease to another. And in that way, what we aim to achieve is an amplification or a multiplication of all of our efforts to benefit many more diseases and many more patients over the next 10 years.
Thank you to everyone for very thoughtful questions. And thanks to our speakers for sharing your insights. I will now turn it back over to Dr. Stamler to close us out.
Thank you, Diana. for facilitating the Q&A session. I'd like to conclude our time together today by reminding you that although the diseases we have discussed today are individually rare, they are collectively common. Rare diseases collectively represent about 1/3 of human disease. About 400 million people worldwide suffer from rare disease, and the majority are children. It's sad, costly, and unnecessary. We're helping to fix that. So how can you support the life-saving work of scientists, scholars, and innovators in rare disease? Stay connected with us. Sign up for information and newsletters at our website. Share what we are doing with your friends and your colleagues. We need share of mind, share of interest, share of belief, and share of vision that we can certainly help develop new therapies for patients with rare disease. Thank you.